Hello, I know you very well. I'm Kevin Giovanoni, a neurologist based in London, and I would like to thank you for inviting me again to talk at your MS Update Forum. I have spoken in the past. I'm terribly sorry that I can't be with you personally in Cairo, which is one of my favorite cities, simply because I'm actually at a meeting in Poland. Anyway, I'm going to go through briefly the immunology around the role of B cells and why we want to target B cell therapies in MS and give you some insights into the future. I hope I can do this all in 20 minutes. I will put this online so you can download the slides. And I am assuming a little bit of basic knowledge around the immunology of MS. So I'm not going to be going through the raw basics. I'm going to assume that you know a lot about immunology. Just to say a large number of disclosures that you need to be aware of. So the B-cell pathogenesis of multiple sclerosis. We know that B cells are involved in multiple sclerosis simply because we find within the central nervous system these B cell follicle-like structures, oligoclonal IgG bands, and cytokines produced by B cells. And we think that these follicle-like structures are important for antigen presentation in terms of activating autoreactive T cells, and they may be crucial in the pathogenesis of advanced or smoldering multiple sclerosis. Now, when you actually look at multiple sclerosis from a systems biology perspective, and you use omics, be it transcriptomics or genomics or even proteomics, you soon find out that the evidence supports T cells, B cells, and innate immune function in multiple sclerosis. You don't really need the omics. We knew this from looking at the pathology of multiple sclerosis back in the 1970s, for example. And the real thing that brings all three of these cell types together is the major histocompatibility complex, HLA. And so my personal opinion is that MS is really, from a pathology perspective, an interaction between T cells, B cells, and innate immunity. And here I'm pointing towards the microglial component or dendritic cells that are antigen presenting. I already mentioned this particular genomic study that was done by the International MS Genetics Consortium. And when they actually looked at specific cell-specific gene regulation, one of the main hits was CD40. And actually, it doesn't matter what you do, CD40 is an incredibly important, or the CD40 ligand is an incredibly important pathway in the pathogenesis of MS and other autoimmune diseases. And also, again, what links the various cell types is CD40 and T cell receptor engagement with MHC. This is one of the works done by Ben Jacobs in our group. He did a summary database Mendelian randomization study, and what came up in one of the nodes was CD40 and the CD40 ligand interaction. So we think this is a pivotal pathway in driving autoimmunity in multiple sclerosis. Now we can target this, obviously, because, as you know, that in most immune diseases, not only do you need an antigen-specific signal, but you also need a co-stimulatory signal. And the CD40 ligand is one of the critical co-stimulatory danger signals that tells a T cell to become reactive, either to fight an infectious organism or to mount an immune response to a vaccine, for example. And we could potentially block this now with an antibody. And some of you may be aware that frexalimab, an anti-CD40 ligand, is currently in phase 3 development as a treatment for various subtypes of multiple sclerosis. And this blocks the interaction between the so-called autoreactive T cell, the B cell, dendritic cells, and macrophages. In other words, it prevents the danger signal or the co-stimulatory signal, and therefore it energizes or switches off the T cell. So it reduces the inflammatory response, hopefully suppresses the autoreactive T cell response, and also reduces the humoral response, the immunoglobulin response, which may be important in MS. You know, whether or not autoantibodies play a role in MS is a moot point. And we've already written up the phase 2 results, just showing you that we're using high-dose frexalimab, the 1200 milligram IV every 4 weeks, reduced new number of new gadolinium-enhancing lesions by over 90%, and this is the dose going forward in the phase 3 program. The question I'm asking the MS community, because theoretically by blocking this T-cell antigen presentation, the T-cell interacting with the antigen-presenting cell, you may be able to tolerize the T-cell and put it into long-term senescence, for example, or become tolerant. And the real question is, when you don't know the specific autoantigen in MS, is how do you, first of all, define immunological tolerance in the MS scenario, and how would you look for it from a research perspective? And that's a conundrum or a problem we haven't resolved, and I haven't resolved either, even having thought about this intensely for months and months, or actually I should say for years. Now, anti-CD therapies are very effective in multiple sclerosis. So this dates back now almost 20 years. And this is the publication from Steve Hauser's group, just showing you that when you use rituximab, and this is the 2,000 milligram dose every 6 months, it was very effective at switching off relapses and MRI activity. This drug never went forward in development. Ocrelizumab went forward, and you know that ocrelizumab is now licensed based on the OPERA-1 and OPERA-2 studies and the oratorio studies to treat relapsing MS and primary progressive MS. So I think there's no doubt that a B-cell depleting therapy targeting the anti-CD20 antigen is a very effective anti-inflammatory therapy in multiple sclerosis. I say anti-inflammatory because despite it being one of our most effective class of therapies at suppressing relapses and MRI activity, it doesn't stop people getting worse. And we can see that people on ocrelizumab, and this is the post-hoc analysis of the OPERA-1 and OPERA-2 studies, almost 90% of them continue to have worsening disability as a result of this phenomenon called progression independent of relapse activity, or PIRA. And so what you're doing by suppressing relapses is you're exposing this the real MS, in my opinion. That's what MS is. You're exposing the neurodegenerative component, which then drives disability worsening in the majority of patients. And that's why our focus now in the MS space is to target smoldering multiple sclerosis. Now, there are a whole lot of pathogenic processes that we think drive this. We think maybe persistent demyelination, innate immune activation, the so-called hot microglial hypothesis, the intrathecal B and plasma cell response, in other words, the antibodies that are being produced, free iron that are driving slowly expanding lesions, energy deficits. This is probably related to mitochondrial toxicity. And I'm in favor of potentially a untreated CNS viral infection driving the disease. And there's also this thing called microscopic focal inflammatory activity. In other words, the fact that we don't see focal lesions clinically with relapses on MRI, we may still be getting small focal inflammatory activity occurring that would need to be addressed by a more potent anti-inflammatory therapy. And this is why we are moving into a phase now where we are trying to develop targeted CNS penetrant therapies. And you will almost certainly have heard about the BTK inhibitors. I'm very excited about CAR T cells targeting B cells, and I'll talk a bit about later CNS penetrant anti-CD20 monoclonal antibodies. A second generation dihydroorotate dehydrogenase inhibitor, vidofludimus, is quite exciting because it seems to be more CNS penetrant than teriflunomide. And we probably have one already, cladribine. Cladribine, as you know, is a small molecule inhibitor that predominantly targets B cells. And we now have quite an extensive data set showing you that it's targeting some of the processes driving smoldering MS. This is work done by my colleague, David Baker. When we actually analyzed the licensed disease-modifying therapies, we found that they all had an impact on the memory B-cell compartment, either reducing them or blocking their trafficking. And the exception being teriflunomide. Teriflunomide doesn't seem to have a major impact on the number of memory B cells in the periphery. The corollary, two drugs, atacicept. So this is actually a drug that blocks BAFF and APRIL. These are two growth factors for B cells. The trial failed because it actually increased disease activity. And similarly, anti-TNF therapies, infliximab, a monoclonal antibody that blocks TNF, also increased MS disease activity. And both of these drugs increase memory B cells. So here we have the corollary therapies, immunotherapies that increase memory B cell numbers and activity, make MS disease worse. Okay, and therapies that block or reduce memory B cell actually make the therapy work. And I actually think this is the therapeutic target in multiple sclerosis. Now, teriflunomide may be working via the B cell mechanism, and one of our hypotheses is that because the memory B cell is the cell where Epstein-Barr virus resides, it may be working as an antiviral, and we've shown in a small study that it actually reduces salivary shedding of Epstein-Barr virus. So teriflunomide may be working via EBV, but not directly suppressing the numbers of memory B cells, but suppressing EBV latent lytic cycling. So yes, MS is a complex disease. We know that all three subtypes of immune function interact with each other, and I think what brings them together with Epstein-Barr virus, which I think causes MS, is the B cell. And we know that EBV is really important. I won't go through the epidemiology. We know that you have to have the virus to get MS, and there's overwhelming evidence suggesting that people with multiple sclerosis do not control the virus very well. And there's an immune dysregulation in relation to Epstein-Barr virus that people with multiple sclerosis have. And I'd urge you to read our review that's out there. I've also got a recent review in Brain from last year where I discussed the therapeutic rationale of trying to target the virus with antivirals. So EBV and memory B cells. So two hypotheses here. One is to try and develop an EBV vaccine to prevent people being exposed to the wild-type virus, and the other one is to try and treat infectious mononucleosis, and hopefully we could stop the cascade driven by Epstein-Barr virus infection. And these are experiments that will hopefully happen in the next decade or two. And I'm hoping in my lifetime we will see a reduction in the incidence of multiple sclerosis as a result of EBV vaccination for primary prevention, or possibly using antiviral agents to treat IM as secondary prevention. Now, coming back to targeting CNS, plasma cell, and B cell biology. So this is an old review now, but it's still very relevant of the types of therapies we could use to target CNS resident plasma and B cells. And on top of the list would be cladribine. The high-dose anti-CD20 therapy failed. The two trials were essentially negative, although hidden within those trials is a subgroup of patients that responded. Those with GAD-enhancing lesions at baseline did much better on high-dose than low-dose anti-CD20 or standard-dose anti-CD20. We've mentioned the CNS penetrant BTK inhibitors. There's proteasome inhibitors. We're doing a trial of exosumab, CD19 targeted CAR T cells, and potentially anti-CD40 ligand may also have an impact on intrathecal B cell biology. Just to say to you that the BTK inhibitors may be working by Epstein-Barr virus. So as you know, the B cell requires signaling by 2 or 3 pathways to remain alive. So if you're a memory B cell, you have to receive a trophic signal, okay, coming via your B cell receptor. And that signals via this complex that involves BTK. Okay, so you actually have to signal via BTK for providing a cell survival. And now, what's critically important that the latent membrane protein 2A, which is a latent protein that EBV puts into the membrane, that bypasses B cell receptor signaling and gives the B cell a pro-survival signal. So this is one way that by blocking BTK, you actually might remove the LMP2A signal that EBV-infected B cells get and kill them. And I hypothesized years ago, actually, that BTK inhibitors are potential anti-EBV drugs. And there is some early data or existing data on the first generation BTK inhibitor, ibrutinib, showing exactly this, that they do reduce latent lytic cycling and they kill memory B cells or B cells infected with EBV. So I personally think that if some of these BTK inhibitors get through the pipeline, we need to also think about them in the context of Epstein-Barr virus driving MS disease activity. Now, the big excitement in the field of autoimmunity is anti-CD14 targeted CAR T cells. So this paper launched the field in a sense. These were 5 patients with drug refractory lupus, nephritis, really end-stage lupus, and they treated them in Germany with CD19 targeted CAR T cells. And the remarkable thing is how well these patients did. in terms of their disease activity scores. They almost went down to zero. And I think the really important thing is their proteinuria cleared up and their complement levels recovered, and they all lost their anti-double-stranded DNA antibodies. It's remarkable. It's almost like a autoimmune cure. Now, some of these patients have remained in long-term remission, in inverted commas, potentially cured, and one patient has actually had a relapse, I was told. So it's not 100% effective, but it's at least much, much better than all the other therapies we had for lupus. And for this reason, the FDA have given anti-CD19 targeted CAR T-cell therapy for lupus a fast-track designation because it's addressing a real unmet need. So as you know, CAR T-cells are designed to bypass the co-stimulatory signal. So what you do is you put the antigen-specific signaling. You put an FC fragment that it recognizes CD19, and you also put a intracellular selenium domain. So as soon as it sees its antigen, it activates the T-cell. It's quite a complicated process because you have to harvest T-cells from people with the disease. You then have to engineer them in a laboratory, expand them, come back to the patient, do a depletion cycle, reinfuse them, and then hopefully the CAR T-cells go around the body, find the B-cells and kill them. And we think that CD19 is probably better than anti-CD20 because it targets a much broader population of B-cells. It even goes up to plasmablasts. And unfortunately, when you become a plasma cell, you lose your CD19 as a plasma cell. So we may also need to supplement anti-CD19 targeted CAR T cells with, say, B-cell maturation antigen, which is also expressed on plasma cells and myeloma cells. The problem with that, though, is BCMA is also expressed on other cell types, so the side effect or adverse event profile will be, I think, a lot worse if we're targeting BCMA as well. But there have been some early trials from China with remarkable results, even in MS, patients receiving both anti-CD19 or anti-BCMA targeted CAR T cells. So watch this space. I think we're just beginning to see the progression of this extremely exciting therapeutic strategy. This is just showing you how effective CAR T cells are at depleting B cells. So we have a pre-CAR T cell lymph node staining, CD19, CD20, and you can see after anti-CD20, which is rituximab in this case, you can still see that there are persistent B cells in the peripheral lymph nodes, whereas CAR T cells completely repeat them. So we think that CAR T cells are much better than monoclonal antibodies at depleting deep tissue B cells, and also CAR T cells get into the central nervous system, and we know that from treatment of CNS lymphomas, they're effective at getting across the blood-brain barrier. So this is why I think CD19 targeted CAR T cells are much more exciting as a B-cell targeting therapy than monoclonal antibodies. And we have data on this. So this is now looking at EBV viral loads in patients and with patients with CNS lymphomas. And I'll just give you two case studies. You can see the CSF blasts. You start the CAR T cell therapy and they drop dramatically. Okay. And on the right, you can actually see patients with CNS lymphoma receiving CAR T cell therapy. And not only are you seeing a drop in the cells, but you're also seeing a drop in the EBV viral loads, suggesting that this is a very effective anti-EBV agent as well. So this is one of the most exciting therapeutic strategies, I think, going forward. Now, what about, I mentioned to you frexalimab, anti-CD40, CD40 ligand targeting monoclonal antibody, but I'd just like to mention that it may also be priming working by EBV mechanisms. So we do know that EBV creates a CD40 signaling pathway analog, LMP1, which is another latent membrane protein, signals via TRAD and activates NF-kappa B pathway. And by blocking CD40, you may actually be removing the co-stimulatory signal that's required to keep memory B cells alive. And it also is interesting, EBV-infected B cells appear to actually express CD40 ligand rather than just CD40, and this may be another mechanism how frexalimab may be working, and may be working via B cell mechanisms. And we think frexalimab may be an antibody that actually affects memory B cells that are EBV infected, and we will be studying this from a biomarker perspective in the frexalimab phase 3 program. So to conclude then, I think B-cell targeted therapies, particularly those that are EBV infected, are critical for treating multiple sclerosis and possibly putting MS into long-term remission and hopefully curing the disease. There's no reason why, from a therapeutic strategy point of view, we may be able to purge the body of EBV infected B cells. In other words, the pool of EBV that drives MS disease activity. And I'd like to make the claim that all effective MS therapies target B cells, okay, in particular the memory B cell compartment. And this is possibly how they're working via Epstein-Barr virus. And I could spend a whole day talking to you about each individual disease-modifying therapy and how they're working via Epstein-Barr virus mechanisms. So I'd like to stop there. Thank you. I will put this talk up on YouTube for you to listen in your own time. If you want to ask me questions, it's probably best to ask me on my YouTube channel, because below we can have a question and answer dialogue. And I'd like to thank my team at the Blizzard Institute and at Barts Health for supporting me over the last 2 decades, where we spent a lot of time thinking deeply about multiple sclerosis and working on mechanisms of how disease-modifying therapies work. Thank you very much.